Now just are we going to start now? I'm going to just read the name of yours. Alexander, yeah, so you were the responders. I just gave you a question last. Dr. Alpina Kairunitsa, okay. And then Dr. Andrian Utomo. Okay. Dr. Su, nice to see you. Okay. Dr. Enda Indriani Wayono. Dr. Esther Devina. Yeah, Professor Fatima. And Dr. Hilmi the Aliani. Okay. Dr. Megawati Megantoro. Dr. Miras Heidi K. And then 우리 학생들, Dr. Nora Frasetio. And Bandung Classroom. And also Dr. Priska Olandari. And Dr. Puspa Handayani, Dr. Septi Cardina, and Dr. Stefanos Masora, and then Dr. William Korompis, Dr. Ulan April Nani, and 김준영 선생 들어왔네. Okay. So, unlike my previous announcement that I'm going to use only 20 slides, I just increased the hidden slide was just exposed. I think this is the right time for me to just give you basic facts. Some of them are from the recent literature, and I'm not responsible for the fact, but it's been already evaluated by the Science or Nature on those editors and reviewers. So we can just depend upon them. The first one is this one. For this one, I'm going to just explain. For half a century, human brain was believed to contain about 100 billion. Last week, I just told you, based upon the reference in science, the 86 billion neurons, but they just said 100 billion neurons and 1 trillion glial cells. But you know, here, glial cell number is only around the 100 billion, not trillion. Please don't exaggerate the number of glia. And it's from the comparative neurology. And the first paper was from the PNAS, Proceedings of National Academy of Science of USA. So I have just here referred in my the review paper, so that I needed to understand the brain. So the first thing is just how many glial cells and how many neurons. Why? Because now we can just image it using the transcriptomics or whatever. So that's here. Equal number of neuronal and non-neuronal cells make the human brain isometrically scaled up primate brain. So that's kind of a reverberation of the evolution theory. And the search of the true numbers of neurons and glial cells in the human brain. Review of 150 years of cell counting. So now they could reach a kind of a little bit the asymptotic goal. Okay. So now it's 1 to 1. But let me just give you, there is a cerebrum and cerebellum. And let me invite Dr. Andreas Lim. You can unmute yours. And then would you please guess? I don't think I can just ask you the right correct question. We are interested in the guess. Let me just tell you, cerebrum and cerebellum. The first answer is, here is a neuron and glia. First one is neuron and glia here, 10 to 1. And in cerebellum, likewise, 1 to 10. The neuron and glia. The second one is 1 to 1, and second choice is 1 to 1. The third choice is 1 to 10, 1 to 10. Which one is correct? Sorry, 10 to 1. The 1 and 1. You can just say anything. We cannot hear you. So while you're preparing yourself to be heard, we repeated second time the Korean Society of Nuclear Medicine Annual Congress last Friday. I was also frustrated and disappointed by the fact that they are using webinar. I really hate webinar. So only the person who are talking with each other is talking each other, has the kind of capability to say, to discuss, and then all the others are the audience. That's kind of, that represents the current great divide between the developed countries and developing countries. It is not going to make the nuclear medicine viable and prosperous. It is not going. It's for the survival of nuclear medicine. In my opinion, in my bias, we need to make it even. Okay, Dr. Andreas Lim. Yeah. So you would have given me the answer, chatting answer here. Where is it? All right. Let me just see. You're chatting. Where is the chatting response? Oh, I think, here. Well, I don't find the Dr. Andres. So it's okay. Why? Because it is the technical problem. But you know, the Korean the IT technology can allow almost 500 people connecting at the same time. Why? Because the comedy program in the television, and also other kind of, we call it Lanson concert. Even BTS can just hold a concert, and then everyone can watch. Yeah, there is no, yeah, it's mainly a comedy program. Anyway, so let me just give you the correct answer to save time. In cerebrum, neuron is 4. No. Neuron is 1 to glia is 4. And in cerebellum, neuron is 4 to 1. That's a crude one. And so that's the reason why the glia would be 10 times. And they just kind of ignore the cerebellum. That might be one of the reasons why we have seen every time the cerebellum is hot with FDG uptake, hotter than the HMPO spec, blood flow spec. Anyway, the second question might go to, I'm just calling you only by just taking advantage of the alphabet order. So Andrean Uttomo, can you just kind of unmute yourself?
Yes, Prof.
Yeah. So my question is, here, I just wrote down, as I just explained, CSF lymphatic outflow in human is the same as rodents, except for the direction. So meaning, am I going to ask you the lymphatic outflow? No, it's a new information. So I'm going to ask you the flow direction of the vein, systole sinus vein or transverse sinus vein. Is it going from frontal to occipital, meaning the anterior to posterior, or posterior to anterior? Do you know the venous flow?
Yes, I think it's going from posterior to anterior.
From posterior to anterior. So, do you have any the reason to think posterior to anterior?
Because venous is from the other part of the body, they flow through the spinal to the posterior and then to the anterior.
Yeah, so it's because of the kind of using gravity, right? But humans are the upright walking animal, so it's different from the mouse. Okay. So you are saying the posterior to anterior. There was a two choice, and then the next question is also based upon your speculation. Then what about lymphatic? Is it the same direction or the counter direction? What is your expectation as speculation?
You right, it is counterflow to the venous.
All right. So yeah, so I just already wrote there. So this is the correct answer. Oh, sorry. Here, one person, cool. Maybe the Asian heritage, 2018. Meningeal lymphatic vessel flow runs countercurrent to venous flow, superior systole sinus of the human brain tomography. And they do just kind of a pulse chase. And then white and black is saying, so now you are looking at, for the first time, the lymphatic flow direction here. At first, it was a white, black and white. Now you would have been quite impressed. Okay, and then unfortunately, human venous drainage, because humans are upright, it might have just kind of a posterior to anterior to posterior. It's okay. However, in case of mouse, they are just kind of a prone position, not supine, prone position. So then heart is the posterior. So anterior to posterior, venous drainage was confirmed. And primates and humans are using the same direction. So lymphatics are coming to the anterior. So then, let me just move on. Dr. Atika Larasaki, can you just unmute yourself? She is not. He is not here. Atika. No, Dr. Chao Su, would you please unmute yourself?
Yes.
Okay, yeah. Then,
yeah.
In mouse, there is known the lymphatics are drained into the nasal cavity. Why? Because the CSF is just above the cribriform plate. So the lymphatics can just kind of easily come down nasal cavity and then nasal cavity to superficial the lymph node of the head and neck. So do you expect humans are using the same drainage site, even though vein is drained to anterior to posterior, and lymphatics are drained from posterior to anterior? Posterior to anterior, then the lymphatics are going to find a way to outside the dura, to the system circulation. So do you expect humans are using the same the route to nasal cavity like mouse or rats? What's your speculation?
Yes, I think so.
Yeah, you think so? Everyone had expected, including my colleague in Daejeon, the Korea. They published their paper in Nature last year, or the 2019 or 2 years ago, and they just discussed that way. However, according to Norway and Sweden report here, cerebrospinal fluid tracer efflux to parasagittal dura in human, parasagittal dura here. Yeah. And then in vivo assessment of CSF efflux to nasal mucosa in humans. In this report, the title was, unlike mouse rodents, humans do not use nasal mucosa as the lymphatic outflow pathway. You know, we are doing nasal package CSF rhinorrhea study in our textbook in nuclear medicine. If people are using nasal mucosa as a normal outflow tract, then CSF rhinorrhea could have been difficult to detect using the posterior packing and then the cotton count, the gauze counting, the study. Dr. Su, do you remember the kind of studies described in the textbook of nuclear medicine, correct?
Yes, correct.
So there is a CSF rhinorrhea and CSF otorrhea, that kind of trouble. CSF rhinorrhea, even though rare. So I suspected that unfortunately, unlike rodents, humans would not use nasal cavity for CSF lymphatic efflux. So it was the case by this group, Mellin, Ide, Ringstad. They reported in vivo assessment and they conclusively found that the CSF efflux is not taking place to the nasal mucosa in humans. That's the reason why our CSF rhinorrhea study worked. So it's kind of really, and then most recently, they have also moved on to the CSF egress, a human parasitical dura, and then impact of sleep deprivation. Interestingly, in Alzheimer's disease, there is a sleep disturbance. Many patients are suffering from the sleep disturbance, and sleep disturbance is a detrimental to the progress of Alzheimer's disease. That's another the point of view of the CSF lymphatic. Thank you for your speculation. Okay, yeah, so please mute yourself, and then I'm going to move on. Next to fact 2 is the most recent one. And then going on to next to fact 2, I just draw something here. Brain has a capillary system. Everyone knows that. Blood is here, running here. As I told you, there is a perivascular space, very narrow space. And in very narrow space, there is the innate immune cell, perivascular macrophage. And perivascular macrophage, their gene expression pattern is the same as the leptomeningeal macrophage. And outside of the here basement membrane, here, there is a famous immune cell, microglia. And anatomically, I would say that there is no anatomical immune barrier. However, there is a immune, not anatomical, but immune barrier is still there. Meaning once inflammation is here, the cytokines are opening the blood-brain barrier, and then many inflammatory cells can just occupy, like in multiple sclerosis. And then once that kind of inflammation is calming down, then here resident microglia are going to expel those the infiltrating macrophages, and then they are taken care of. This is kind of military. This is police. Brain is like that. However, most recently, there has been another similar phenomenon here. Synovial cells are best known as a kind of a target of the autoimmunity in rheumatoid arthritis. And once rheumatoid arthritis flaring, then the many monocyte-derived macrophages are here and causing problem. In addition to neutrophils, we know that the neutrophils are the worst aggressor. But once the activity, acute phase has been just kind of reduced, has been passed, then synovial cells are going. So they just explained like this, they just explained this area, there is an immune barrier. The synovial cells and then stromal, the cells, stromal tissue cells of the joint is another example. So this kind of functional, the barrier is there. However, there's no absolute, the anatomical barrier is there. No anatomical barrier is there, but functionally they are just a kind of a sanctuary. That's the reason why both the not true, but true. It's just kind of another fragmented expression derived from Korean language. So please excuse me. And then fact 2. And fact 2 is going to discuss with Dr. Su. You can just unmute. Ah,
Yes.
yeah. So yeah, good. Yeah. So Dr. Su from Myanmar. And let me just first explain to you. The last month in Science, there has been a paper here. Heterogeneity of meningeal B cells reveals lymphopoietic niche at CNS borders. Meaning, skull has a bone marrow, and skull has a channel. And just beneath the skull, there is a dura. Yeah, dura here. And then that skull has a direct channel with a dural vein. And dural vein does not have any tight junction. So cells can escape the dural sinus, venous sinus. And then they just kind of can make even lymphopoietic niche. The authors would be and best expert interested in mainly the multiple sclerosis. So they try to find B cells, how B cells can just kind of be exposed. We all know that the multiple sclerosis, the myelin basic protein is a autoantigen, and then myelin, there are autoantibodies. So we barely know that why, because the multiple sclerosis is not a major target in nuclear medicine. However, we just kind of heard the rumor that those B cells, systemic B cells, are not a problem in case of multiple sclerosis. CSF B cells, B cell clones would be a problem, the most recent report. Anyway, we have bone marrow there. Skull bone marrow is also bone marrow, and bone marrow has a direct anatomical connection. And then they could just say that using the single cell analysis, the skull bone marrow is a major source of the B cells here, and then it is going to be make a difference. And then here, the second fact is lymphatics here. Skull and vertebral bone marrow are myeloid cell reservoir for the meninges and CNS parenchyma. So, Dr. Su, would you please mute a little bit while after this presentation? I would just Dr. Septi Hardina, please mute. Yeah, thank you. And here they just kind of plainly saying that the skull bone marrow is the source of the myeloid cells to the parenchyma. Isn't it astonishing? So 6 years ago, nobody knew that dura has a lymphatic channel. Now, after 6 years, 2021, in another science paper, the brain's myeloid-derived cells are not from the systemic bone marrow, but from the skull bone marrow. So until now, until reading this paper, based on my medical school lessons acquired from my professors at that time, I thought that the bone marrow is the same from skull, another flat bones like scapula or pelvic bones. Those are flat bones. They're keeping the bone marrow until the end of the day, meaning until the elderly, the epoch. And then when we were so young, the long bones, even the distal end of tibia fibula, and also the not humerus, but radius and ulna. Okay, it's a long time since I just read the bone scan. So So radius and ulna, however, once the subjects are reaching the adult age, only the 1/3 of humerus and 1/3 of the femur has bone marrow. So vertebral bone marrow and skull bone marrow and other flat bones bone marrow are have been considered as one compartment. This paper refuted that idea. They have irradiated the mouse's body in a separate experiment and mouse skull. And then they have connected two mouse together using circulation. One is fluorescent animal, the other is one. Finally, long story short, they have found the same channel from bone marrow to dura, via dura, spinal cord. And they are saying that here, vertebral bone marrow is taking care of the spinal cord. Skull bone marrow is taking care of the brain. So that's a kind of astonishing phenomenon. When I just read for the first time the 2015 Nature paper saying that, oh, there is a lymphatic in dura and there is some way from CSF to lymphatic, is totally different from what I have learned when I was in medical school. So it was really interesting, and I decided to propagate this idea, and I to present this idea to Warmth meeting, to Vietnamese the invited lecture, and try to just now convey to all ASEAN next generation leaders, colleagues. So Dr. Su, let's move on to here. Yeah, so at first it's showing this one. So there was 3 years ago, there was a good paper, and this is a movie. The pathologist colleagues, they are making this kind of fancy imaging. Okay, and then here is a nanosinus. And then these are channels. Okay, slap. Seeing is believing. Actually, the nuclear medicine people, we are visual people. We don't like to listen to anything. We like to see something, and we'd like to show the definitive functional anatomy to our, actually functional physiology to our colleagues. And we are the best. And yeah, yellow. Skull marrow meninges connection. At that time, in Nature Neuroscience, there was a really, and why I just put Kim is a Korean in one of the private university. Dr. Kim is a neurologist. Neurologist is interested in this kind of, and we need to just kind of accumulate the knowledge, at least not so later than our neurologist or psychiatrist or neuroscientist colleagues, to collaborate with them. Maybe once cancer or tumors are conquered completely, the brain might be another frontier. Why? Because currently, brain has frustrated a lot of people. And this is the my drawing here. Let me just show you again. Skull has a bone marrow, and then just under the skull, periosteum would be there. There is a dura there, and then the dura has a sinus. And I just showed you parasitial dura. And then green colors are lymphatic, which was found in Nature paper. And then just beneath the dura, there is an arachnoid. It's a really membrane. And that membrane consists of the arachnoid barrier cells. And this is a tight junction. And CSF and the outside of brain is controlled by the arachnoid barrier cells, ABC. And arachnoid barrier cells has a arachnoid, spider web type of a loose tissue. And along the loose tissue, CSF is running. And CSF is running this ABC and PR surface. PR is taking care of the integrity of the parenchyma. And just beneath the PR, there is an astrocyte end foot process. That end foot process is making syncytium. Syncytium, once called syncytium, now reticular. Why? Because it is not really one cell. It's just kind of a combinations of multiple cells, astrocyte end foot process. And then PR artery is poking through here, and PR surface is running and then stop somewhere. And that vessel, around the vessel, perivascular space there. So this kind of complicated structure is now can now be understood like this. Skull marrow, parasitical dura is connected, and parasitical dura and lymphatics. Lymphatics has a sinusoidal epithelium, so has a many pores, and veins too, so they are connected. And then if there is any connection across the arachnoid barrier cells, then the CSF and the dural space can be connected, and then leptomeningeal and perivascular space is automatically connected. So brain is infiltrated alongside the perivascular space into the deep side by CSF. That's the way how the CSF floating cells can reach the brain parenchyma, just to kind of passing the PR surface. You know, the PR surface does not have any tight junction. PR surface has a gap junction, which is much more easily penetrable instead of the blood-brain barrier. So it's not surprising that recently CSF is the kind of a good reservoir for treating brain tumor. They do not inject systemically because blood-brain barrier is a hurdle. So they do just kind of to treat a meningeal seeding, they do inject the anti-cancer chemotherapy drug to intrathecal route. Now you understand. CSF is a very good reservoir. So, Dr. Su, I'm going to give you a simple speculation question. According to my explanation, which is least explained pathway? So everything is now well explained. For CSF should escape from CSF space to dura across arachnoid. So is it well explained?
No, I don't think so.
Yeah, so you are correct. So thank you for your kind of correct answer.
Thank you, sir.
Yeah, I think I have explained the completely for the first time. I try to convey this knowledge to my lab members, and to my residents during the morning conference, and they simply could not understand me. And then why? It's because, you know, I did not explain. I explained all my drawings except for P. Fisher here. You see Fisher, Dura Fisher. This one and this one. Arachnoid barrier cell has a tight junction, should have some channel to route to just kind of deliver CSF to Dura or Dura to CSF. In recent PNAS paper, they found a Fisher structure here. So actually, arachnoid barrier cells has a holes. That's an interesting phenomenon. Still, this report was not recapitulated. However, in Nature paper 2015, they just draw this, and arachnoid villi was like this one. Here, arachnoid was protruding to the sinus, inside the sinus, and then it was easy to assume that this might be the very good path. But in addition to this one, there are Fisher structures lined by the lymphatic endothelial structure. So now I finished my my persuasion for all of you. Brain is not an anatomical sanctuary. Brain is an active organ to communicate with the systemic immune system, but not the entire bone marrow. Not vertebral bone marrow is taking care of the spinal cord. The skull bone marrow is taking care of the brain. That's the most novel information that I can give you today. So I have almost consumed I have invited three or four of you to just take part in, and this would have, I hope this would have just encouraged you to study immune drugs like PD-1, PD-L1, or other drugs, and then some of you would be involved would participate in finding the brain tumor treatment, which is now fatal, almost fatal, and also neurodegenerative disease. That's the reason why I just spent a lot of time to you, and why? Because it's kind of a paradigm shift. If one paradigm is the sustaining, there is no way to just escape from them. I was one of the victims of the amyloid tau paradigm. I really hate that kind of waste of passion and waste of. So now fact 3. Next one would be Edison. If you just can, and the next one would be Dr. Enda, Dr. Edison, Dr. Esther. Yeah. So if I recognize your name as among my professor colleagues list, then I just invite him or her. The fact 3. Now you can just a little bit. You can be. Get a little bit relaxed. I'm going to just kind of give you some information, and then last 30 minutes, meaning the around the after 30 or 40 minutes, I'm going to use another 30 or 40 minutes to give you the facts. Now, why? Because you are already enlightened by the novel facts, which was organized and then amalgamated by me. And then that novel fact is everyone knows that the region layer specific neurons. Okay, and Harvard neuroscientist Xiaowei Zhuang, Chinese American woman scientist. She's the best in this field, and she used to she tried to spatially resolving and making cell atlas here. Layer 1, 2, 3 is and for the mouse, these layers are shallow, thin, 5, 6, 7, and in human, these layers are like this. So that's the difference between rodents and humans. And so there are glutamatergic and GABAergic and non-neuronal in situ hybridization. So this kind of information is had been just a basic information, but interestingly, this can be replicated here. Glutamatergic L6B neurons are here, but it is not specific this layer. The amygdala has the same the gene signature, and here habenula thalamus has the same signature, and here glutamatergic L5 layer is here, L5 here, and then retrosplenial cortex has the same signature, and that kind of information is from transcriptomics. And then what about the region specificity? So we used to segment our data based on MRI. So I can say that the century of MRI is gone, and another century or decades of transcriptomics. This is a diagnostic imaging. This is pathology. We need to just kind of graduate from diagnostic imaging. We need to just integrate our knowledge with pathology. Let me show you. Here, the PhD student as a fellow, he did a marvelous job. He just did this kind of analysis, and then he just also adapted the data from Harvard, and then analyzed this one. This is just a segmentation. 12 animals' brain was segmented by their transcriptome pattern. Globus pallidus is a little bit weird and promiscuous. It means the globus pallidus transcriptomes are shared by many areas. However, striatomystus, caudoputamenistus, and isocortexes, thalamus is only to the thalamus. But, however, hippocampus is shared, gene pattern is shared by many the isocortical areas. So now we need to just understand the neurons and in each spot. You can think that this is a voxel, but that voxel is a size is like this, 100 micron, 100 micron, 10 micron. Very crude as a pathology. However, much better than our imaging. FDG PET, we have now 4 millimeter, 4 millimeter, 4 millimeter. MRI, 1 milli, 1 milli, 1 milli. Still better than this one. But the shortcoming is just one section. If you have a lot of money, then you can have several sections, but it takes so much money. Okay. Now, Dr. Edison. Then let's come to our own discipline. Dr. Edison. I don't know. I'm just saying to you the FDG PET. Here the human. Dr. Edison, can you unmute? Oh, you cannot. Then what about the Dr. Enda? Can you unmute? You know, I'm also checking our capability of interactive discussion. So far, almost half of our colleagues can communicate. They can just open their own voice. All right. Then let me just move on. Dr. Hilmi, Aliana. Sorry, Aliani. Can you? All right. So then, Dr. Gil, can you unmute?
Yeah, I'm hearing.
Yeah. So FDG PET. After listening to my just presentation, can you say now this part of the FDG uptake is from oligodendrocyte, and this part is from neuron, and this part is from astrocyte, and this part is from microglia. So my question is yes or no, and then why? Or why not?
My answer is no.
Then why not? Why not? May I just give you the chance to use our previous one? So this is the condition of the brain. Astrocyte and oligodendrocytes and even neurons. And neurons are supported by astrocyte and their synaptic clefts are the nearby, and then astrocytes are the third party to take care of the glutamine, glutamate recycling and whatever, and the kind of issues. 우리 분자약 바이오작과의 대학원생들은 제가 얘기하고 있는 바이올로지컬 팩트를 의사들한테는 중요하지 않지만 여러분한테는 이거 여러분이 할 수 있어야 되는 겁니다. 물론 여러분이 뉴로사이언스 필드에 연구를 한다면. 그러니까 now, Dr. Gil, would you please explain the answer is no, and it's obvious. So explain why it's not an easy task. However, you can just improvise your answer.
I guess some region have some dominant type of specific cells, but all the regions have all the type of cells, I guess. So FDG uptake is not only because of the specific cell type, but also because of all the type of cells.
Then my following question. Would you please explain why 20 years ago you did not consider to become the nuclear medicine physician? You did not know anything about FDG PET. 20 years ago, why 20 years ago, best expert in the United States, UCLA, who had been invited to Korea to give us a lecture on the dinner table, an assistant professor, an ambitious assistant professor, but frustrated to take care of the everyday routine, who is Dr. T.S. Lee. When I asked him, FDG PET in brain, is it representing neuronal glucose uptake, and why? He said yes. So do you mean no, meaning this is also neuron, this is also neuron, this is also neuron? 20 years ago, everyone thought like that. FDG PET is from neuron. FDG uptake is neuron. FDG uptake of the brain is representing neuron. They were so sure. Why? Yes, you are correct. Meaning, absence of answer is a correct answer. You don't know why. Okay?
Yeah.
But you know, I have gone through that kind of big folly, decade of big folly, and there has been the hot dispute. Washington University has produced Peter Fox, who's been in San Antonio after that, and then UCLA has produced Heine Sherbert. And Cernin, the current, and Marcus Schweiger. And they were all involved in the muscle imaging, meaning muscle and tumor imaging. Peter Fox was the brain guy. And there was that expert was Heine Sherbert. Heine Sherbert was confident that no, it's and then they they annoyed Peter Fox, saying that brain is different, and there is a lactate shuttle. He said lactate shuttle, and astrocyte and neurons are collaborating. And around that time, 20 or 30 years ago, there was no information regarding the presynaptic and postsynaptic astrocyte kind of coupling. Glutamate does not have, the neurons does not have any glutamate transporter. Glutamate transporter is only in astrocyte, and astrocyte is making glutamate, making glutamate to glutamine here, and glutamine does not have any charge. So glutamate is easily transferred from astrocyte to, again, it's different from dopamine. Dopamine neuron is the reuptake site or dopamine transporter. That's the reason why dopamine transporter imaging, DAT scan is possible to find out the postsynaptic neuron from here, where midbrain substantia nigra is going to the striatum, and then the axon is here. Axon terminal is postsynaptic site, and here neurons are receiving. They are on button on the dendrite, and then that's postsynaptic. Presynaptic terminal has a dopamine transporter. Dopamine transporter is imaged by FPCIT, as I told you. and F18 was devised, adopted by the Professor Kim, and also recently, IDAN 123 FPCIT. Previously, it was 123 beta CIT. Now, all of a sudden, you listened to the further explanation in addition to Dr. Son and Professor Kim previously. Now, that's the situation. So in one voxel of the brain, there are so many cells. This is different from the here. In heart, everything is muscle. Sometimes endothelium, sometimes fibroblasts, sometimes the immune cells infiltrating. But in brain, so much convoluted with each other. So, and we all know that the lipopolysaccharide, if given, these are microglia cell line, not macrophage, microglia cell line or a primary culture. Oxidative phosphorylation is down and glycolation is high. The PhD student, Dr. Seo, could just to make this and to understand this phenomenon in transcriptomics domain. So this kind of the innovative work can be done as a PhD thesis, but not for a clinical purpose. So let's move on to the next. Technical tips. As I told you, 100 micron, that spot has 200 million identifier. One spot has a 1 to 10 cells, average 5. Among 22,000 genes in humans, and 200 are variable genes, differentially expressed in analysis, dozens to hundreds marker genes can be acquired. So the spot has a count, the 1 to 5 in some the rare genes, and popular genes, and 20 to 70, or much rarer genes, 1/10 or 1/100th, 1/2. So after total Rees normalization, logarithmic transformation is done. So all these all these numbers, digits are going to become 0.1 to 5. So that's log normalization. And our Affymetrix machine using they are doing this kind of reads. That's the technical issue. You don't need to understand or remember. But interestingly, this is again, tissue section can be analyzed. And then each tissue section's counts can be interpreted or predicted using what? The deep learning algorithm. When I had been visiting Bandung, and from Bandung to Jakarta on the train, I had several questions. And at that time, the resident, the chief resident doctor asked me how Korea is integrating IT technology with this one, not inventing that, but just adopting the estranged area, meaning me. I belong to clinical. I'm just interested in a little bit of math and technology. But you know, there is a kind of 30 years ago, we could not do this. To do this, we need to go to the United States or the Europe, mostly the North and West Europe. But now this technology is everywhere. Monetary support is another problem. Okay. So that's the issue. And recently, astrocytes were found to be layer specific, really deeper and the upper. However, it's a little bit different from brain. However, striatum enriched one and hippocampus enriched astrocytes are a little bit localized. And mouse brain could be layered according to the neurons, and also layered a little bit differently according to the astrocytes, their gene signatures. And once they are given the microglia growth receptor blocker, they call it PLX compound. Then once removed, microglia can regenerate on site, not from bone marrow. And how many are going to take care of the vicinity or not, what kind of receptor is going to work or not, was reported, and which is beyond our expertise. And there had been a report, microglia, their status can be classified like this, and then even the quantified absolute amount in hippocampus or thalamus. And their homeostatic microglia also increase in neurodegenerative disease. Disease-associated microglia is increased. Then what's the difference? So that's a current question. So I'm trying to understand. I don't think the world leaders in this field understand. They just do because there is a new technology. And amyloid plaque-associated microglia too. What's the difference? So please don't be disappointed that you are behind. While you are listening to my explanation, you are at the cutting edge. Not everyone should do this kind of research, but we can just listen to the others' experience. Okay, and then I'm going to move on. So, and there has been various reports, and I just checked if there is a similarity between reports, and then a little difference, but and then a little bit difference, grossly the same, but in detail a little bit different. So 5 years in science and 3 years later in Nature Neuroscience, they are saying the same thing. So we can just understand. And so as Dr. Gill has responded, then what are we seeing in our everyday meeting? Glycolysis and oxidative phosphorylation are done by every kinds of cells in the brain. And FDG uptake is showing just the glycolysis. Actually, FDG uptake is showing glucose transport activity convoluted by hexokinase activity. And that's, we simply say that it's glycolysis. Then is it by neuron, astrocyte, microglia, or others? So now I'm going to just ask, I'm going to take a survey after Dr. Gill. Dr. Megawati, can you unmute Okay, thank you. And if you just kind of say nothing, then I would interpret it as a technical difficulty because so far we were so familiar. We got too so familiar with our kind of unilaterality of information transfer. Dr. Miraz, Hedy, sorry. Can you unmute? I'm not going to ask you the fact. I'm going to ask you your opinion. Okay, Dr. Miraz too cannot. Why? Because this is here in Korea, it's just after 2 o'clock, meaning the afternoon, but in the morning, they are so busy in taking care of the routine work. So I'm so appreciating your participation. Dr. Nora Prasetyo.
Yes, Doc.
Yeah, so now I just explained a little bit. So brain has some area has a high uptake. Now, how much percent of confidence or conviction you can say it is by neuron? So I'm not just asking you the correct answer. I can just let you know there is no correct answer now, currently. But well, I'm just kind of asking you your speculation. How much some voxel has a high FDG uptake? How much of the FDG uptake is, so how many voxels among the total voxels are from neuron? That's one kind of interpretation, but I'm just, I have tried to convince you that our thinking should be based upon the unit by unit, voxel by voxel, sometimes region by region. So one can say the frontal lobe, one can say it's a hippocampus, that kind of amygdala, or the lateral temporal lobe, so neocortical area. So I'm just asking you, randomly you just pick one voxel, and that voxel has a high uptake in FDG. So can you say it's a neuronal uptake or just astrocyte uptake? And if you say it's neuronal, then how much percent of confidence do you have?
Neuron, maybe. About 70.
Pardon? How much?
Neuron, Prof.
How much percent?
Maybe 60, 70.
60, 70%. Okay. So as I just told everyone, we don't know the answer now. So in that sense, in that case, previously it was 100% neuron. The 30 years ago, if I say, according to my just a study or literature search, it can be from microglia, then I would be just kind of expelled from the community by just not evidence-based, speculation-based research. But now the Dr. Nora Prasetyo, she just said percent. So the question was because, and thank you for your answer. And And the question and answer is to explain, to find out this or not this, or 20% or 30%, which one is correct? What kind of information should we know? So at first we need to know that in one the spot, how many are neurons, how many are microglia. And then we need to know that neurons are consuming how much of glucose is glycolysis. And then glycolysis can be represented by FDG uptake or the glycolysis related peptide or glycolysis machinery. They call it machinery. 20 or 30 genes are known to be involved in glycolysis, just like hexokinase and glucose transporter. And then even pyruvate dehydrogenated complex and pyruvate carboxylase and phosphoenol pyruvate or something. And you might just remember all those enzymes. And that kind of enzymes can represent FDG? No. So, however, we need this kind of information. And then what about astrocyte? Astrocytes are the working as presynaptic and postsynaptic astrocyte. That's one unit. Without astrocyte, glutamate cannot be handled and excitotoxicity is taking place. That's happening in penumbra zone. Penumbra, ischemia, during ischemia, because the circulation is blocked and they are just kind of in miserable condition. So glutamate is not taken care of. Dopamine is high. Then it's a kind of a drug addiction or internet addiction or game addiction, that kind of situation, cocaine. And however, glutamate high, then it's a kind of a toxicity. So astrocytes are actively involved in the metabolism here. And how much of the metabolism is being taken part of, is being done by astrocyte is not known now. It should be quantitatively understood, quantitatively. And we have FDG PET. FDG PET is a huge amount of glucose is coming in, but we use a very small amount of F18 FDG. And then if here, inward and outward is stationary, then FDG PET is going to give us the, even using lumped constant, and the difference of the deoxyglucose and the glucose, that difference is taken care of the lumped constant. And even we can just find the glucose metabolic rate, cellular metabolic rate of glucose. This is the strength of our FDG PET, but we don't know neuronal contribution, astrocyte contribution, and recently microglia contribution. So that's the end of the second part. Why don't we just take a 5 minutes break and then coming back, and then use our remaining 15 to 20 minutes for just wrapping up. That wrapping up is for the wrapping up is dedicated to what? Clinical ones. Just kind of a reiteration of the information which was not emphasized by Professor Kim Yu-kyung, Professor Kim, first two lectures, and the Dr. Song, the most recent lecture. regarding epilepsy and cerebrovascular disease and AD again. I'm obsessed with Alzheimer's disease. So now 26, 31, 2:31, we will resume. And stay tuned. Please don't go away. Please don't turn the channel. Thank you. See you soon. As we are going to start 2 minutes after, so let me just pronounce the participants after Nora Prasetio. So Dr. Stepanos Masora, Dr. Tu Nguyen, Dr. Tu Trang, and Dr. William Kromkis, and Dr. Julendri Xiaoqi. Okay. And then, yeah, that's all. So sorry not to invite for question and answer. So you don't need to be concerned about your possibility to be incorrect. No, my question usually do not have any answer. It's just kind of asking your opinion. Okay, 31 to 31. Let me just summarize. And the first one is cerebrovascular disease. What's the difference between HMPAO and ECD? So I just drew. The water PET for cerebral blood flow using O15 water, C11 CO2. C11 CO2, was it? No. I was wrong. It's because, no. It's because, where is it? I was wrong, and this was not O15 CO2. O15 CO2 is that O15 is interchanging with H2O. So it's actually CO2 imaging is water imaging. And then the brain blood flow. And then here, there is an assumption stationary with flow. Then flow is stationary, then large K1 and K2. That's volume of distribution and partition coefficient. And I tried to explain this last year at the beginning of our lecture series. So I'm just kind of repeating. If you have any questions, then please, you can ask me, or you can just ask Professor Jae-sung Lee to explain in more detail in our winter workshop. His lecture is going to follow after next week is the Professor Seo, and then the next week, next to the next week, 2 weeks after, last lecture is going to be by Professor Jae-sung Lee. He is going to explain in a most easiest way. He's among physicists and also among nuclear medicine physicians in my department. He's a really good lecturer. He's doing a lot of cutting edge research, but he gives his lecture in a very understandable, comprehensible way. So oxygen PET, cerebral oxygen metabolism, oxygen 15 O2 is regional cerebral metabolic rate of oxygen, is oxygen contents of the hemoglobin and hemoglobin amount, and their saturation, that this kind of hemoglobin amount, and one hemoglobin can just carry how much amount of oxygen content, and multiplied by saturation 90% and 70%, then people are hypoxia. And then cerebral blood flow, and then oxygen extraction fraction. And that kind of issues are going to make the similar lambda or partition coefficient, and OEF can be understood like that. And then this kind of information follows. This kind of information, or what is this? And previously, according to Dr. Song, vasodilation is taken care of, and then OEF is taken care of later. This is wrong. According to this paper, this kind of compensation takes place simultaneously at the same time. So that's first, second, and third is wrong, according to 20-year-old report. And that's the reason why the blood flow decrease at rest and flow reserve cannot be matched like this. There is this kind of reserve abnormality is there. Okay. And we know that when parts of our brain is activated because of the contribution of PET, 20% is increased blood flow. And the CBF is increased up to 50% by what? Drop. CO2 inhalation is also increased. That kind of basic fact had been discovered by PET. We are PET experts. Even though you did not do any the study using PET, you are also PET experts. Why? Because we are the only person who understand the earlier the articles or review papers and textbooks written by our ancestors, the nuclear medicine pioneers. And then, what's the difference between HMPO and ECD? It's like this. HMPO is now stable. They don't care. No, not this one. We don't care. And then ECD is representing not as well as RCBF convoluted by esterase activity. Actually, this is not a flow agent. If some the pharmaceutical company agent is saying it is going to promote ECD use, no. HMPO is good. That's my idea. However, the many things are in common. So because we are image reading experts, you can use ECD. But let me just tell you, in addition, beside this kind of difference, as ECD image is a little bit inferior, less contrast. Contrast. Lesion to contralateral. Okay, so, and then what kind of UCG triage? Here, adult, everything is ambiguous. 80s, around the 20, around the turn of the century to millennium and 2010, there had been 3 times of the controlled trial. They did not find any difference between therapy. In their court, they did not use diamoxpect. So my colleagues are using, my colleagues means neurosurgeons, are doing actively the diamoxpect. What about the Moyamoya disease in the young? 30 years ago, very young, was risky. Operation was risky. So they sent every one of the patients to us after 25 years. Now, their operation risk is low. And behaviorally improved, angiographically improved, and more frequently we followed up and improved. They now know that everyone is going to improve by just looking at the child. So they don't do this one. They don't do this one. This one was a definitive benefit and no risk. And just angiography and then operation. But in case of the symptomatic adults with hemorrhage or even asymptomatic adults, this belongs to here. They are still doing the astrocytes spect. That's one slide conclusion of my trial of 26 years of diamoxpect. Now, epilepsy. Which radiopharmaceutical? Around 2000, turn of the millennium, we just switched it for 3 months to ECD and found this kind of case here. Here, definitive. Here, a little bit the blurred. And we found that the asymmetric index was different. Professor Song has given the slide, so I just throw. Why? Because this is the slide that I made almost 20 years ago. In some cases, ECD is normal and HMPA is abnormal. So my neurologist and neurosurgeon colleagues hated ECD. So we do not use ECD. So just to, please don't follow our track, but just to, you can just find. And neocortical epilepsy is a difficult one and rare, but difficult. FDG PET and ICTAR SPECT helped. Why? Because some of the one is, they call it non-regional or cryptogenic, meaning all MRI normal. MRI normal, then that's cryptogenic. Even temporal lobe epilepsy is cryptogenic, meaning here, hippocampus does not show increased the signal, meaning no sclerosis, and volume is normal, no atrophy. Then our neurologist and neurosurgeon colleagues are helpless. Then what to do? ICTAR SPECT or PET, and then cryptogen, and then ICTAR SPECT and ICTAR EEG. ICTAR EEG is dura is removed, and then grid is inserted, and then dura is again the sutured. So using this one, 5 to 10% of subdural hemorrhage. This is a serious complication. So they try to avoid this one. And once a lateralization is done, they can just insert not both sides, but this side. If they say that frontal, then they just can use only strip instead of grid. So we did a lot of good job, and now the current scheme of the triage is hippocampal sclerosis, and the atrophy is then no other, and then directly hippocampal amygdalectomy is done, cryptogenic, and then do that. And based on this one, and 80% or more is improved, 87%. And neocortical epilepsy is not, is a really the resilient fall. So as I told you, subdural, if you can just help them, the full lateralization. So several years ago, they have followed up 10 years, in some cases 20 years. And they found that PET localized cases and SPECT localized cases, 20 years. 20 years are really good. So that's a textbook knowledge. And maybe the last slide. Professor Jae-sung Lee and now Professor Hong-Yun Choi, they are doing AI or deep learning approach. Jae-sung Lee is doing the try to remove MR, try to remove CT, and then simultaneous. PET is enough. Why? Because the attenuation map can be drawn using transmission, emission. We don't need transmission scan. Emission image is also containing, and that kind of information was successfully for the first time in the world. Now everyone is doing similar approach. So this is not because of this kind of progress is not because of we have Samsung, we have IT. It's because we have Jason Lee, and it's because we have MD, but deep learning expert. So you can do that. Not now, but within 10 years, I think we can do that. Right, so 247. So this is a real good summary, and you can, I would have consolidated the knowledge which was given by Professor Yu-Kyung Kim, but I could not do anything about the Parkinson's disease. Why? Because I do not know anything about Parkinson's disease. And the further, a deeper understanding of amyloid and tau imaging. It's kind of amyloid imaging, amyloid and tau story, and lack of the the therapy after following up 30 years had traumatized me. So, which is going to be objectively by Professor Saul. And biomarker story too. I don't know very much. So Professor Saul is going to follow me in according to his decision, he can at first the correlative imaging of lymphoma and hepatoma and FAPI, and then he can collect the questions from you. Interestingly, I asked questions, and you gave me enlightenment. You're speculating similarly to me based on this kind of information. So I could be sure that my understanding could be shared. You can just kind of deviate from my bias to make your own story. That's good. That's kind of really wished, really desired by this kind of interaction. Okay. Thank you. And as I just consumed so much time, I think the next semester is going to be coordinated maybe mainly by Professor Seo. I can just kind of volunteer 1 or 2 occasions, but mainly by Professor Seo. And Professor Seo would need your contribution, meaning we have a full list of 220 once or more time participants list. So we can just ask you and how to make it. And even one of you can volunteer. I'm going to say this thing, and then I'm going to just take feedback from my colleagues in Asian countries. Why not? It's yours, and I think I and Professor Seo would have successfully constructed a platform, and then until no one has any technical difficulty in unmuting him or her, is going to be and then I'm going to remove unpublished material from my slide, just like the previous one. It will shrink a little bit. And then we'll just also upload to our homepage. Then it is CC, Creative Commons. You can just download. And you can use the remind yourself, or you can use it to deliver your own the understanding to deliver to your juniors. It is more than allowed, meaning it is highly recommended. And if you find something wrong after 1 or 2 years, please let me know. Then I could just change my the understanding. Professor, so do you have any comment? Any announcement?
No, I currently don't have. Actually, next week I am planning to discuss about firstly about radiation biology, but nextly about I would like to have some interactive survey of what for the next semester actually.
Good. Yeah. So that's kind of asking questions. Okay. So please they help us and let this kind of a platform to be a help to you, not for us. Okay. Thank you. Bye.
교수님.
Thank you very much.
Thank you, Professor Lee.
Yeah. Nice to see you again, Professor Hendra.
교수님 혹시 들리시나요?
네, 들려요. 네.
저 질문 하나만 하고 싶은데.
네, 누구죠? 길 선생.
저 길준영입니다. 죄송합니다.
네네. 네.
그러면 RI 시스토노그래피를 하게 되면 결국에는 CSF랑 클림파틱 둘 다 뭐 보게 되는 거라고 할 수 있을까요? 다만 그 두 개를 구별하는 게 그렇게 큰 의미는 없는 것이고. 그렇게 이해를 하면 될까요?
지금까지는 그 안에 서큘레이션을 봐서 지금 목적이 그거였거든요. 프리피션트 하면
맞습니다. 네.
딱딱 그거 하나예요.
네네네.
네, 내가 저 학회 때도 잠깐 얘기했지만 김재승 교수인가 양승호 교수님인가가 아주 오래전에 인트라크레니얼 하이포텐전이라고 해서 그 CSF 리노레아 오토레아 아닌데 새는 게 있다고 그랬거든요. 언더 프로덕션하고 새는 거하고 둘 중에 하나거나 둘 다잖아요.
네네.
그래서 새는 거를 보여준 적이 있어요. 그러니까 그렇게 하려면 브레인에 있는 브레인하고 스파이널 코드에 있는 액티비티 다 가려야 돼. 그래야 렉이 보이지. 그렇죠?
네네.
근데 우리 저 아스말이 발견한 거는 저 옵트레이터 파라버테브랄 이런 것도 발견했는데. 그리고 저 서민석 교수 아직 있나요?
네, 있습니다.
그거 저기 만약에 지금 저 하게 되면은 좀 많이 주면 어떻게 되는지를 한번 보면 좋을 것 같아요.
많이 준다는 게 볼륨을 말씀하시는?
네, 볼륨입니다. 네.
네.
예, 그 concentration, 그러니까 혹시 스페시픽 액티비티가 낮게 나오면은 실험 준비해놨던 거를 그냥 버리지 말고 스페시픽 액티비티 억지로 낮게 만들어 달라 그러면 박지용 박사가 모랄 해저드에 빠지거든. 그래서 계속 높게 만들어 달라 그러는데 낮게 만들어 주면 잘 됐다고 그러고 6마리를 통째로 왕창 줬더니 다 죽었더라. 뭐 그럴 수도 있고 죽지 않았으면은 도대체 너무 많이 주면 혹시 림프노드로 안 빠질까 봐. 프레셔가 높아지면서 전부 코로 다 빠져버릴까 봐. 그런 거죠. 그래서 지금 하여간 뭔지 모르지만 하여간 그동안 숨겨져 있던 거를 얘기를 했습니다. 오케이, 마칠게요. 그럼